Alright everyone, so this video covers the last two sections of chapter 13. Feel free to rewind or speed up this video if you need to, or pause it at any time. Obviously you're going to do this on your own pace. Today we're going to talk about techniques that you can use when you apply the other techniques that we've learned about in previous chapters. So after you've been able to study the DNA and analyze it and all this stuff, you can use that information in order to do other things with it. Okay, the two techniques that we're going to talk about today is cloning and creating a transgenic organism. Now, cloning is the idea that you can make an exact copy of something. So if I wanted two of myself for whatever reason, I would make a clone of myself. And you've seen a lot of this on TV shows and movies where a person, walks into a machine or whatever, and all of a sudden there's another copy of them walking out or what have you. That's not exactly how it works, unfortunately. Now, in order to clone something, you're going to start with just a single cell, and you're going to allow that to divide until it eventually forms a full organism. So if you were to clone yourself, you would need one cell that contains your DNA. You will cause that cell to divide, and eventually, after your clone is born, it will grow up and it will become you. But that takes a while, right? You can't just have something give birth to a full-grown animal or organism. Not yet, at least. The first successful cloning was done in 1997 when a scientist named Ian Wilmot was able to clone a sheep named Dolly. And this was a huge breakthrough for science because his techniques were pretty simple but relatively successful. I mean, he did try hundreds and hundreds of times in order to make one successful clone, but the technique that he used has since been replicated and duplicated by other scientists to clone other animals, such as dogs or cats or mice or what have you. Now, there is a benefit that is seen to cloning. For example, science has always wondered, well, could we use this technique in order to save, for example, an endangered species? If there's only a few hundred of them left in the wild, could cloning possibly save that species from going extinct? On the other side of this debate is, well, is this ethical, especially when we're talking about cloning humans? Should humans be allowed, or should we allow humans to be cloned? Because if that technology becomes successful, some people are afraid of possibly having something like the Clone Wars, just like in Star Wars, right? Where you make a bunch of clones and you send them into war instead of I guess you quote unquote normal or regular civilians. Okay, so super soldiers, things like that. We're not too sure on where that's going to lead, and it is a very gray area. Here's how cloning works, and here's how you make a clone. You basically start off with the egg from a female sheep. So this sheep on the right-hand side is the original egg donor. So we take one of her eggs from her body and we remove her DNA from the egg. Because remember, sperm and egg cells only have half the number of DNA. And in order to clone something, you need to have a diploid cell. So we're going to take the DNA out of the egg, and now it's just an egg without any DNA in it. We're then going to take DNA from a different sheep, from the sheep that we want to clone. This fully white sheep here we're going to call Dolly Senior. Okay, so you take any diploid cell from the animal or organism you want to clone, you take out its DNA from that cell, throw away the cell because you don't need it, and you're going to put that DNA into the empty egg cell. You're going to fuse them together. To fuse means to glue or to attach to each other. After you've been able to fuse the egg and the DNA together, you're going to allow it to divide. You're going to form what's called a culture. Okay, a culture is just a clump of cells. You let it divide for a little bit, because when you implant it or when you place it into a surrogate mother, you want to be able to make sure that it's at least starting to divide, is successfully dividing and starting to form the little baby. Okay. Now, a surrogate mother is basically any animal or organism that is carrying the baby that's not hers. So this sheep, for example, is the surrogate sheep. It is carrying the embryo, or the early stages of a baby is called an embryo. It is carrying the embryo from a that contains the DNA that is from a different sheep. So this is not her baby. This is somebody else's, right? Humans do this too. We also have surrogate mothers that will carry babies for others, give birth to the baby, and then the parents will then claim the child, right? So this sheep here is called the surrogate, and she will carry the sheep until it's ready to be born. Um, and when she gives birth to the sheep, it's going to be an exact clone of the sheep that you took the DNA from. Okay, so DNA from Dolly Sr. was put into an egg from a different sheep. It was then allowed to culture and then implanted into a surrogate. And when the surrogate gave birth, she gave birth to Dolly. That finishes up cloning. The other technique that we're going to talk about is creating what's called a transgenic organism, which is an organism that has DNA from two different species, okay, or two different sources. So in order to do that, we go through a technique called transformation. Transformation, if you guys remember from previous chapters, is when you take the DNA from one organism and place it into another. Just like Frederick Griffith did with the DNA from one virulent bacteria that he added to the DNA of another bacteria that was non-virulent, and he was able to change it. He was able to transform it. To transform means to change. So when you have DNA from something else inside of you, you are going to change. Okay, you're going to transform. It is quite easy to do in bacteria, and we do this using recombinant DNA. Remember, we talked about using restriction enzymes, and that recombinant DNA is when you take DNA from one organism and another and you put them together. To make a recombinant DNA, you cut both pieces with a restriction enzyme, and to see whether successful or transformation was successful, meaning that the bacteria was able to pick up that piece of DNA or pick up the transformation, you're going to check by shooting with a bunch of antibiotics. If after giving this bacteria a whole metric poop ton of antibiotics, it doesn't die, then chances are transformation was successful. There are a lot of drugs that we can make doing this. Human growth hormones is one such thing that we do this. We have also created insulin using this technique by putting human insulin gene into, I believe it's cows, and that will allow us to make a transgenic organism that can create insulin for us. And of course, you guys hopefully understand or know by now that that's to treat diabetes or to help with diabetics. Now, in order to transform bacteria, we need to use little pieces of circular DNA called plasmids. Okay, so plasmids are naturally occurring, and they are great for transformation for two different reasons. A plasmid will actually have a piece of DNA sequence in it that tells the cell that it's in to make copies of itself. Okay, it promotes its own replication, in other words. So once you get a plasmid into a bacteria and that bacteria starts reading that DNA, it's going to want to make copies. And that's a good thing, because once you've successfully transformed a bacteria, you want to have many copies of it to test to see if what you were hoping for is true. Okay. Now, yeah. Blah. Secondly, plasmids are great for transformation because it has genes on there called genetic markers. Genetic markers allow us to test to see if that bacteria has been successfully transformed. One such thing is, or one such genetic marker is antibiotics resistance. Okay, so there is a gene inside of the plasmid that allows bacteria to be resistant to antibiotics, meaning that if you give yourself antibiotics and it's one of the bacteria that has that gene, you won't be able to kill that bacteria. Okay. Um, if the bacteria was successfully able to pick up that plasmid and use it, then you should see that it doesn't die when you add antibiotics to the mix. So let's see how this works. Okay, so in this picture we have two different pieces of DNA. We have your plasmid, which is the bacterial DNA. It is a round piece of DNA. And on that plasmid, there is a gene that allows bacteria to become resistant to antibiotics. So this yellow part right here. This over here is DNA from the organism or from whatever it is that you want. So let's say this is the DNA for, I don't know, for bioluminescence, the thing that allows fireflies to glow in the dark. Let's say this plasmid, you want to make bacteria that glow in the dark so that you can see where it's dirty. I don't know. Okay, so here is the gene for bioluminescence. You want to cut both the plasmid and that gene with the same restriction enzyme, creating those sticky ends. When put together, the sticky ends should allow the plasmid to stick to the piece of DNA that you want. Sometimes it doesn't always happen, but sometimes it will. Once you've figured out which one has been successfully turned into a recombinant piece of DNA, you're going to then take the recombinant DNA and you're going to try to place it into a bacteria. Now, this isn't always successful. Sometimes you just have to take the recombinant piece of DNA, place it into a test tube or a beaker with the bacteria, and hope that the bacteria picks up that plasmid. If the bacteria was successful at picking up the plasmid, it should now have that little yellow gene that allows it to become resistant to antibiotics. Okay, so it's also going to have that gene that you want for glow in the dark, for example. But it has that little yellow gene because it was successfully transformed when it picked up the plasmid. If that's the case and you spray it with antibiotics, it should live and then it should continue to make copies of itself. If the bacteria was unsuccessful at picking up the plasmid, was not transformed, for example, like this bacteria here, when you add antibiotics, the bacteria will most likely die. We can use a similar technique to help transform plants. Once you've transformed bacteria, you can and you can use that bacteria to help transform plants. Now, there is a naturally occurring bacteria that causes plants to produce little tumors within their tissues. One of the things that science has realized is that when the bacteria infects the plant, it's going to take a piece of its own DNA and insert it into the DNA of the plant. This causes the tumors to form. When scientists were able to figure out which part of the DNA and which gene the bacteria inserted into the plant, they used this to their advantage. What we did was we took a piece of DNA and we placed it into the exact spot where that gene is, that gene that allows that the bacteria, is placing into the plant to create tumors. So we cut that gene apart, we placed a new piece of DNA in there, created a recombinant piece of DNA inside of the bacteria, and when we infected the plant with that particular bacteria, it's going to try to put that piece of DNA into the plant for us. Okay, so the bacteria doesn't know that it's been transformed, that it's been changed. It just knows that this piece of DNA needs to go into the plant so that I can create tumors. If you replace that piece of DNA with what you want, the bacteria will place that DNA in the plant for you. It's pretty sneaky, but pretty darn smart. Other times, in order to transform a plant, all we have to do is remove a little bit of the cell wall, and sometimes that plant will take up the DNA on its own. Okay, so I don't believe it's as successful as doing the bacterial technique, but I could be wrong. Again, using this technique, you can make a transgenic organism. Transgenic organisms are organisms that contain DNA from other species in them. So, for example, plants that glow in the dark have firefly DNA, or the antifreeze tomato has fish DNA, or that papaya from the video last Friday had viral DNA inside of it. So that is what a transgenic organism is. We can make bacteria that produce human growth hormones, tobacco plants that glow in the dark, etc., etc. The list goes on and on. Okay, one of the things that we use a lot in genetically modifying foods or GMO technology to modify food, is to produce plants that have natural insecticides inside of them, meaning that if an insect were to eat this plant, it would die, right? And in order to do that, we take DNA from other organisms, from let's say plants and insects that are poisonous to other plants or to other insects, and we place it into that plant, and so now that plant can produce that insecticide. It's one of those gray areas that people are still unsure whether that's a healthy thing, but it's something that we can do. One of the last things that I'm going to talk about is making a transgenic mouse. Lab mice or lab rats have always been used for study. And if you are able to create one that has a very similar uh, immune system or very similar genes to humans, you can study how drugs might affect us if it were to be given to humans. Okay, so there's an ethical issue there as well for animal rights and what have you. Um, but since you can't really test on humans due to even bigger ethical issues, it's, I guess, one of the quote unquote better alternatives. Lastly, this is a picture of a plant that glows in the dark. I took it straight from your book, and it's one of those new technology things that we've been looking into for possibly reducing the amount of energy that is being used by us to illuminate things at night, or whatever. Okay, so that ends the video on 13.3 and 13.4. I hope you guys were able to get all the information, and if you have any questions, email me or ask me in person, and I'll try to do what I can to explain it.